Welcome back. It feels like we've barely dipped our toes in this whole world of preclinical pharmacokinetics.
It's true. There's always something new to discover, especially when we're talking about how drugs work inside the body.
And I'll admit, sometimes it feels like learning a whole new language. With all these terms like volume of distribution and elimination half-life, it's a lot to take in.
I understand. It can be overwhelming at first, but each of those terms has a purpose. It gives us crucial information, like how the drug moves, how long it stays active, and in the end, how we can make sure it's used safely and effectively.
It's like a doctor checking those vital signs, right? To see how a patient's doing.
Exactly. And just like doctors need to understand those vitals to make the right treatment decisions, researchers need these preclinical parameters to figure out how to design clinical trials.
So before we jump into the world of human testing, should we do a quick recap of those key concepts from this season, just to make sure we're all on the same page?
Absolutely. It's always good to have a solid foundation, especially when we're dealing with drug development.
So let's start with volume of distribution. VEED. I remember you describing it as this imaginary container that holds the drug.
Yeah. Imagine a drug dissolving in like a container of water. The VEED is like the size of that container you'd need to get the same concentration of the drug that we see in the blood.
But it's not a real volume, is it? It's more of a way to understand how much a drug spreads out through the body.
Exactly. It's a way to quantify a drug's tendency to go into different tissues. Some drugs, like amphetamine, have a really large VEED, which means they easily distribute into fatty tissues and organs.
Amphetamine has a huge VEED, right? Like 200 liters. That's bigger than any person.
It is. And that actually tells us something important about how the drug works. It affects how long it stays in the body, how likely it is to build up in certain tissues, and even how well it can cross the blood-brain barrier.
So VEED isn't just a number. It really tells us a lot about how the drug behaves.
Exactly, and that's why researchers pay so much attention to VAID. It helps them predict how a drug will be absorbed, distributed, and eliminated, which is all essential info for designing safe and effective doses.
That makes sense. Now, what about elimination half-life? That one always makes me think of a ticking clock.
Yes. The T half. This one's a bit more straightforward. It's the time it takes for the body to get rid of half of the drug dose.
So if a drug has a half-life of, let's say, 4 hours, that means after 4 hours, half of it is gone from the body.
Precisely. And this is crucial information for figuring out how often a drug needs to be taken. A drug with a short half-life, you might need to take it multiple times a day. But one with a longer half-life might only need one dose.
It's all about finding that balance. The drug needs to stay in the body long enough to do its job, but not so long that it builds up to dangerous levels.
Exactly. And just like with VAID, a drug's half-life can be affected by a lot of things. Age, how well the liver and kidneys are working, even other medications.
So it's not always a set number. It can change depending on the person.
That's right. And that's why personalized medicine is becoming more and more important, tailoring the treatment to the individual patient.
That's fascinating. So we have VEED telling us how the drug spreads out and T telling us how long it sticks around. But what about the actual process of getting rid of the drug? That's where clearance comes in, right?
Right. Clearance, or CL, is essentially a measure of how well the body removes a drug from the bloodstream. It's like a filter.
So a drug with high clearance would be like a super efficient filter,
Yeah.
quickly removing the drug from the blood.
Precisely. And different organs contribute to that filtering. The kidneys filter the drug out through urine, which is called renal clearance, and the liver breaks down the drug, which we call metabolic clearance.
Like a team working together to get rid of something that doesn't belong there.
Exactly. And understanding clearance is essential for preventing drug buildup and potential toxicity. It helps figure out the right dose and how often to give it to maintain the right levels without overwhelming the body.
Got it. So we've got VD, TF, and C. Feels like we have the basics down for understanding how drugs move through the body.
Right, and these are the fundamental concepts that lay the groundwork for everything that follows, as we transition from preclinical studies to human testing and clinical trials.
And that's a big step, isn't it? Going from cells and animals to actual human volunteers.
It is a big step, one that needs careful planning and a strong commitment to ethical principles.
So before we talk about the phases of clinical trials,
Yeah.
maybe we should touch on the regulations that govern this whole process. I know we've mentioned the FDA and ICH before, but could you remind us what they do?
Sure. The FDA in the US and the ICH internationally are like the watchdogs of drug development. They set the standards, review the data, and they ultimately decide if a drug is safe and effective enough to be used in people.
So they're the gatekeepers, making sure only the most promising and well-tested drugs make it to the market.
Exactly. And their role is crucial for public trust in the whole process. They make sure the trials are ethical, they're scientific, and that the safety of the participants always comes first.
It's good to know there are such strict safeguards in place.
Absolutely. These regulatory bodies are involved in every step. They review the preclinical data, they keep an eye on the trials as they're happening, and then they evaluate the final results.
Like a constant quality check, making sure everything is done right.
Exactly. And their involvement is absolutely essential for ensuring that the research is done with integrity and that new drugs are developed responsibly and ethically.
All right, so we've laid the groundwork. We've recapped those key preclinical concepts, and we've highlighted the importance of the regulatory bodies. Now, I'm excited to dive into the phases of clinical trials and see how all of this translates to testing in humans.
I am too. It's a fascinating journey that shows us the power of scientific inquiry, the importance of collaboration, and how committed we are to improving human health.
But before we get into the specifics of each phase, I think it's important to talk about the ethical considerations involved in this type of research.
You're absolutely right. Ethical considerations are crucial throughout the entire drug development process, but they become even more important when we start testing new drugs on humans.
It's a huge responsibility, making sure that the rights and well-being of the participants are protected at all times.
Absolutely. And that's why informed consent is a cornerstone of ethical clinical research. Participants need to fully understand the purpose of the study, what's involved, the potential risks and benefits, and their right to leave the trial at any point.
It's about empowering patients to make decisions about their own health and whether they want to be involved in research.
Precisely. And ethical considerations go beyond just informed consent. We need to make sure trials are designed in a way that minimizes risks to the participants, that data is handled responsibly, and that the results are shared in a transparent and accurate way.
So it's a comprehensive approach, upholding ethical principles throughout the entire process.
Exactly. Ethical conduct is key for maintaining public trust in scientific research and making sure the data collected is reliable. It's a fundamental principle that guides every step of drug development.
It's inspiring to see how scientific rigor and ethical considerations go hand in hand to move medicine forward and bring new hope to patients.
It really is. And as we explore the world of clinical trials in more detail, we'll see how these principles shape every stage, from the initial design to the analysis and reporting of results.
I'm looking forward to getting into those details and understanding how all these complex ideas translate into real-world applications.
I am too. It's a journey that highlights human ingenuity, the importance of collaboration, and the constant pursuit of better health.
But before we can completely shift gears to clinical trials, there's one more preclinical concept I want to make sure we touch on: absorption.
Yes, you're right. We've talked about how drugs move through the body, but we haven't really discussed how they actually get into the bloodstream in the first place.
Exactly. That's where absorption comes in. It's how the drug enters the systemic circulation, so it can reach its target and do its job.
Precisely. Now, if a drug is given intravenously, absorption isn't really a factor because it goes directly into the bloodstream.
It bypasses that whole absorption step.
Yeah.
But what about drugs that are taken orally or in other ways?
That's when things get more complex. The way a drug is given makes a big difference in how it's absorbed.
So a pill someone swallows and a drug injected into a muscle would have different absorption pathways.
Absolutely. For oral medications, the drug needs to dissolve first, and then it has to get through the lining of the stomach or intestines to reach the blood.
It's like going through an obstacle course.
Exactly. And all sorts of factors can influence how quickly and how much of the drug is absorbed. Things like the drug's formulation, the person's age and health, even the food they've eaten recently.
It sounds like a delicate balance to make sure the drug is absorbed properly.
It is, and that's why researchers spend a lot of time studying the absorption characteristics of a drug. They need to figure out the best way to give it, the right dose, and the right schedule to make sure it gets where it needs to go safely and effectively.
So absorption is another piece of the puzzle, especially for drugs that aren't given intravenously.
Right. And it's something we need to keep in mind when we talk about clinical trials, because absorption can affect the drug's overall effectiveness and safety in humans.
I see. It's all starting to come together. We've got VD, T-half, CL, and absorption. All these pieces working together to give us a complete picture of how a drug acts in the body.
Exactly. And with this solid foundation, we're ready to move on to the next stage, the exciting world of clinical trials.
I can't wait to see how all of this translates to real-world applications and ultimately helps to develop those life-changing treatments.
Me too. It's a testament to the power of scientific inquiry and our unwavering commitment to better health.
But before we jump into the world of clinical trials, we should talk about the regulations that surround drug development.
Absolutely. We can't just start testing drugs on humans without making sure it's safe and ethical.
That's where organizations like the FDA and ICH come in, right?
Exactly. The FDA, which is the Food and Drug Administration here in the United States, and the ICH, or International Council for Harmonization, globally, they both play a huge role in regulating drug development and making sure that clinical trial participants are safe.
So they're the guardians of the process,
Yeah.
making sure everything is done by the book.
Precisely. These organizations have strict standards and guidelines that pharmaceutical companies have to follow all the way through the process, from those early preclinical studies all the way to when the drug is approved to be sold.
So what are some of the key things they do?
Well, first, they review and approve every single clinical trial protocol before it can even start. This is to make sure that the trials are designed ethically, scientifically, and that they properly protect the safety of the people taking part.
So it's a very thorough quality control check
Yeah.
to make sure the trials are done to the highest standards.
Exactly. They also monitor the trials while they're happening to make sure everything is being done according to the rules and good clinical practices.
So they're not just involved in the planning, but they're actively overseeing things.
Right. They review all the data that's collected during the trials to figure out if the drug is safe and if it's working.
Like judges, carefully looking at the evidence.
Good analogy. And finally, they have the power to say yes or no to new drug applications based on all the data that's submitted.
They make the final call on whether a drug is good enough to be sold and used by patients.
Exactly. The FDA and ICH play really important roles. They make sure that new drugs are developed responsibly and ethically, and that ultimately protects public health.
It's reassuring to know that those safeguards are in place to protect patients and make sure that only the safe and effective treatments get approved.
It is. And as we move on to discussing the different phases of clinical trials, we'll see how these regulatory bodies continue to shape the whole process.
I'm really interested to hear more about those details and how scientific rigor and regulatory oversight work together to move medical research forward.
It's a fascinating partnership. It really shows how important collaboration is when it comes to developing new and innovative treatments for people who need them.
It really shows the spirit of scientific advancement. Researchers, regulators, and patients all working together.
Absolutely. And speaking of patients, let's talk about them for a moment. The people who volunteer to test these new drugs.
It's amazing to think about the courage and selflessness it takes to participate in a clinical trial. You're trusting in science and potentially putting yourself at risk.
It's true. And that's why ethics are so important when we're designing and running these trials. We have to make sure that the participants know all about the potential benefits and risks, and that their rights and well-being are protected at all times.
So informed consent is really important in clinical research.
Absolutely. Participants have to choose freely to be in a trial without any pressure or coercion. They need to understand what the study is about, what will happen, what the risks and benefits are, and that they can quit at any time without any problems.
It's about treating people with respect and making sure they can make informed choices about their own health and their participation in research.
Exactly. And ethical considerations go beyond just informed consent. We also need to make sure that the trials are designed to minimize any risks to the participants, that their privacy is protected, and that the data we collect is handled responsibly.
So there are many layers of ethical considerations that need to be carefully addressed.
That's right. And these considerations are essential throughout the whole drug development process, from those early preclinical studies to the final phases of clinical trials. It's about upholding the highest ethical standards, to make sure the participants are safe, that people trust scientific research, and to make sure the data we get is accurate.
It's inspiring to see that scientific rigor and ethical considerations can work together to help us make progress in medicine and give patients new hope.
It really is. And as we wrap up our discussion today about preclinical pharmacokinetics and the move to clinical trials, I hope you have a better understanding of how complex and important this field is.
It's been a great journey. We started with those technical terms that might have seemed intimidating, like volume of distribution, elimination, half-life, and clearance. And now we understand how they all play a crucial role in designing safe and effective trials.
We've explored the ethics that guide this type of research and seen how scientific curiosity and the commitment to patient well-being drive progress in medicine.
It shows what we can accomplish when we use human ingenuity and collaboration to unravel the mysteries of the body and develop new treatments that can make lives better and even save lives.
Absolutely.
So, as we end this part of our discussion, I encourage you to keep thinking about what we've talked about today and consider this question. What advancements in drug development do you think we'll see in the future, and how will these innovations change human health? Until next time, keep diving deeper. Okay, so we've covered how drugs get absorbed, distributed, and eliminated. But that elimination rate constant, K, still kind of throws me off. It feels like it's working behind the scenes.
You're right. K doesn't get as much attention as elimination half-life, but it's still really important. It tells us a lot about the dynamics of how the drug is eliminated.
I remember you saying that K and T half are connected mathematically. How does that work again?
Sure, the elimination half-life T half is actually calculated from K. It's 0.693 divided by K. So they're really two sides of the same coin, giving us different ways to look at the same process.
Okay, so T half tells us the time it takes for half the drug to be gone, but K gives us a more detailed view of how fast the elimination is happening, no matter how much drug is there.
Exactly. Imagine watching water drain out of a bathtub. T half would be like marking the time when the tub is half empty. K would be more like measuring how fast the water level is going down each minute.
That makes it clearer. So K is about speed, while T is about hitting a certain point.
Precisely. And just like things can affect how fast water drains from a tub, like the size of the drain or if anything is blocking it, a drug's elimination rate constant can be influenced by different factors too.
We talked about how things like age and liver function can change the T half. Do those same things affect K?
Absolutely. Anything that changes how well the body can get rid of a drug will affect both T half and K. For instance, if someone's kidneys aren't working well, they might not filter the drug out of the blood as efficiently. That would lead to a lower K and a longer T half, which means the drug stays in the body longer.
Like a slow drain, right?
Yeah.
The water level goes down slower because something is blocking the drain.
Exactly. And this shows why it's so important to think about each patient individually when deciding on the right dose. Someone with slower elimination might need a lower dose or to take the drug less often to prevent it from building up and becoming toxic.
It's amazing how interconnected all these concepts are.
It really is. And understanding this complexity is what helps us develop safe and effective medications. That's why those preclinical studies are so important. They give us that baseline understanding of how a drug behaves in the body and help us figure out how to move it into human testing.
Okay, so we've talked about absorption, distribution, elimination, and even those rate constants. It seems like we have a good grasp of the preclinical pharmacokinetics now.
We do. And with this knowledge, we're ready to move on to the next stage of drug development, clinical trials.
I'm excited to see how all these concepts actually work in practice with human participants.
Me too. It's an incredible process. It requires a balance of being scientifically rigorous, considering ethical implications, and constantly striving for medical advancements that can make a difference.
Speaking of ethics, we talked about informed consent a bit earlier. Can you tell us more about that? It sounds like a really important part of running ethical trials.
It's absolutely critical. Informed consent is the very foundation of ethical clinical research. It's all about making sure the participants understand exactly what they're agreeing to before they join a trial.
So it's not just about getting someone to sign a form. It's about a real conversation, making sure they understand the risks and the potential benefits, and letting them make their own decision.
Precisely. Participants need to know what the study is about, the procedures involved, the potential side effects, and that they can stop participating at any time without any consequences. They should be comfortable asking questions and raising any concerns they have.
It's about respect, recognizing that they have the right to make choices about their own health.
Exactly. And ethics go beyond informed consent, too. We need to make sure the trials are designed to minimize any harm to the participants, that their privacy is protected, and that the data we collect is handled responsibly.
So it's a multi-layered approach, keeping ethics at the forefront throughout the entire process.
Right. Ethical conduct is crucial for building trust in research and making sure the data we get is reliable. It's a guiding principle for everything we do in drug development.
All right, so we've covered the basic preclinical ideas, talk about the regulations, and discuss the importance of ethics. Now I'm ready to learn more about the actual phases of testing with humans. What does that roadmap look like?
Sure, clinical trials typically have three main phases. Each phase has its own goals and challenges.
And each phase builds on the one before it.
Right, exactly. It's a step-by-step approach to figure out if a drug is safe and effective. We start with a small group and then gradually include more and more people as we go.
Let's start at the beginning then. What's the main focus of phase 1 trials?
Phase one is all about safety. It's the first time a new drug is tested in people, so the main goal is to see if it's safe and identify any side effects it might cause.
So it's like dipping your toes in the water before jumping all the way in.
Exactly. We usually start with a small group of healthy volunteers, and we monitor them very closely for any bad reactions.
Oh.
We also use this phase to figure out the best dose range for the drug, to find that sweet spot where it works well but doesn't cause too many side effects.
So we're setting the stage for the next trials by figuring out the safety and the right dose.
Precisely. And if a drug makes it through phase one successfully, it moves on to phase two. That's when we start looking at how well the drug works.
So in phase 2, we're checking to see if it actually does what it's supposed to do.
Right. This time, we include people who have the condition the drug is meant to treat. We watch them closely to see if the drug makes a difference in their symptoms or if it helps slow down the disease.
So we're moving from safety to effectiveness, getting evidence to see if the drug really delivers on its promises.
Exactly. And while safety is still very important, in phase two, we also get more information about the ideal dose, the best way to give the drug, and any potential interactions with other medications.
It sounds like phase 2 is crucial for figuring out if a drug has the potential to actually become a treatment option.
It is. And if the drug continues to show promising results in phase two, we move on to the last stage of testing, phase three trials.
Phase 3, that's the big one, right? The final test before a drug can be approved for everyone to use.
You're right. Phase three trials are the biggest and most comprehensive. We usually have thousands of participants at different locations. The goal is to definitively prove the drug works, monitor its safety over the long term, and compare it to other treatments that are already available.
So we're gathering strong evidence to get the drug approved and show that it's valuable in real-world situations.
Precisely. We try to make phase three trials as representative as possible of all the different types of people who would use the drug if it's approved. They give us the most complete picture of the drug's benefits and risks, which helps the FDA make informed decisions about approval.
It's a long and demanding process, but it's how we make sure that new drugs are safe and effective before they reach patients.
It is. And while most drugs that enter clinical trials don't make it to the market, the ones that do have the power to change medical care and improve many, many lives.
It really highlights how powerful scientific inquiry is, how dedicated researchers are, and the courage of the people who volunteer to participate in these trials.
Absolutely. It's a team effort. And it's what drives progress in medicine and gives hope to people all over the world.
Okay, so we've outlined the 3 main phases.
Mm-hmm.
Safety in phase 1, effectiveness in phase 2, and confirmation in phase 3.
Mm-hmm.
But are there any other types of trials involved in drug development?
You're right. There's one more we should talk about. Phase four trials.
Phase 4, what happens after a drug is already proved and people are taking it?
That's where phase four trials come in. We do these after the drug is already on the market. The goal is to get even more information about its safety and effectiveness over the long term.
So it's like a constant monitoring process, even after the drug is out there.
Exactly. Phase four trials can help us catch rare side effects that we might not have seen in earlier trials. They also give us valuable insights into how the drug works in different groups of people or when it's used with other treatments.
So we're basically gathering real-world data to improve our understanding of the drug and make sure it's being used in the best way possible.
Exactly. Phase four trials are a crucial part of what we call post-marketing surveillance. They help ensure that drugs stay safe and effective for patients over the long haul.
That's good to know. The monitoring doesn't stop once the drug is approved. It's an ongoing process.
It is. And this ongoing surveillance is really important for catching potential problems early and taking steps to protect patients.
Okay, so we've covered a lot today. Those basic preclinical concepts, the regulations, the ethical considerations, and all of the phases of clinical trials. But I have one more question. What happens if a drug doesn't pass a certain phase? Does it just get shelved?
That's a great question. It really depends. Sometimes, if a drug shows serious safety concerns or it just doesn't work, we stop developing it. But there are other times when a drug might get a second chance, especially if researchers think it still has potential.
So it's not always a dead end.
Not necessarily. We might try changing the formulation, adjusting the dose, or trying different ways of administering it. We might also do more research to understand why it failed and see if there are ways to overcome those obstacles.
Like trying to solve a mystery and find a way forward.
Exactly. Setbacks are part of the process, but they often teach us valuable lessons that can help us in future research and lead to new discoveries.
It's a reminder that scientific progress isn't always a straight line. There are twists and turns along the way, but the ultimate goal is always to improve people's health.
I agree. Every step we take, every lesson we learn, brings us closer to that goal.
Well, this has been incredibly informative. We've come a long way from those initially complex-sounding terms to understanding the entire journey of drug development.
It's been quite a journey. I hope you now have a deeper appreciation for how complex, rigorous, and promising this field really is.
I definitely do. It's remarkable how scientific curiosity, ethical considerations, and the drive to improve health can all come together to make progress in medicine and bring hope to people all around the world.
It really is remarkable.
So before we move on, keep those brains engaged. Think about everything we've talked about and consider this. If you were a researcher developing a new drug, what factors would you prioritize in your clinical trials to ensure both scientific rigor and ethical conduct? It's a tough question, but an important one to think about as we continue to explore the always evolving world of drug development.
It's a crucial question. It highlights how important it is to have a well-thought-out and balanced approach to research. We need to be scientifically strong and ethically sound to make sure new treatments are safe, effective, and available to everyone who needs them.
It's a delicate balance, but it's essential for moving medical research forward and improving human health.
Absolutely.
All right, I think it's time for a short break. We'll be back soon to look at some real-world examples of clinical trials and discuss the challenges and successes in this fascinating field. Until then, keep thinking and keep those questions coming. Okay, we're back, and I'm ready for some real-world examples. I want to see how VID, T, clearance, all of that stuff actually works in a real trial.
It is pretty fascinating to see those concepts in action.
So, where should we start? Got
Yeah.
any good examples in mind?
Sure. Let's imagine we're working on a new drug for high blood pressure. Millions of people deal with that, so it'd be a big deal.
All right, high blood pressure. What's step one?
Well, let's say our preclinical study showed this drug has a pretty long half-life, maybe around 24 hours.
That's a good thing, right? Less pills for the patients.
Exactly. One dose a day would probably be enough to keep the right amount of drug in their system.
And what about the volume of distribution?
Our data suggests a moderate VID, meaning it mostly stays in the bloodstream and doesn't really build up in other tissues.
So less chance of weird side effects in other parts of the body.
Right. And our studies also show that it's mainly cleared by the kidneys.
So we'd have to be careful with people who already have kidney issues. Maybe adjust the dose.
Exactly. That's something we'd think about carefully when designing the trial.
All right, so we know the drug's pharmacokinetic profile, long half-life, moderate VID, cleared by the kidneys. How does all that shape our phase 1 trial?
Well, phase 1 is all about safety, remember? We'd start small, with a group of healthy volunteers, and slowly increase the dose to figure out the maximum tolerated dose, the highest dose before we start seeing bad side effects.
And we'd watch those volunteers like hawks, right? Any sign of trouble, we stop.
Of course, looking for any signs that the drug isn't safe enough to move forward.
And since it's cleared by the kidneys, we probably wouldn't include anyone with kidney problems in this first trial.
Good point. No need to put anyone at unnecessary risk.
Okay, so let's say everything goes perfectly in phase 1. What's next?
On to phase 2. Now we start to see if it actually works in people with high blood pressure.
So we're bringing in people who actually have the condition we're trying to treat.
Yep. And we'd split them into groups. Some get the new drug, others get a placebo, or maybe the standard treatment for high blood pressure.
Controlled trial. So we could really compare how well the new drug stacks up.
Exactly. We'd be tracking their blood pressure closely, looking for significant drops in those taking the new drug.
But we're still keeping safety top of mind, right?
Always. Safety is a priority in every phase.
Okay, let's say the drug does well in phase 2. It lowers blood pressure without causing serious side effects. What happens in phase 3?
Phase 3 is the big test. It's the last hurdle before the drug can be considered for approval.
We're talking thousands of participants, large-scale stuff.
Exactly. We're confirming its effectiveness, comparing it head-to-head with other treatments, and gathering long-term safety data.
Building a really strong case for approval.
Right. We want these phase 3 trials to reflect the real world as much as possible, so we include lots of different types of people who might use the drug if it's approved.
And if everything looks good after phase 3, we submit it to the FDA for approval.
Yep. They review all the data, looking at the benefits and the risks.
And if they decide it's safe and effective, they give it the green light.
Exactly. It's a long process, but it's all about making sure new treatments are safe and effective for the people who need them.
It's amazing to see how all this works, from the early research to actually getting a new treatment out there.
It really is. Science, ethics, and human ingenuity all working together.
And it all starts with those core concepts we talked about. V-Day, T-half, clearance, absorption. They're the foundation for everything that comes after.
Absolutely. Understanding those concepts is key for designing effective trials, working with the regulations, and ultimately getting new treatments to patients.
Well, this has been an incredible deep dive into drug development. It's inspiring to see how it all comes together to improve people's lives.
I agree. It's a testament to the power of science and the dedication of everyone involved.
As we wrap up this deep dive, think about what we've learned. And consider this. What do you think the future of drug development holds? And how will those innovations change the future of human health? Until next time, keep on diving deeper.